For those of you that don't know who we are, we're a clinical stage stem cell therapy business. So what we're trying to do is harness the power of stem cell science and convert that into therapeutic products, therapies that patients can have administered to them to alleviate very substantial unmet medical needs. And I'll come on to what those are in a second. Importantly, what we do with stem cells is produce allogeneic treatments, and that means they are non-patient specific. So rather than taking the patient's own stem cells and manipulating them in some way and then reintroducing them, what we're looking to develop is a standardized off-the-shelf product, if you will, that any patient can have administered to them who presents with the disease in question. So we're not reliant on the patient's own stem cells. That's the autologous approach, which certainly has merit in a number of therapeutic settings. But for the diseases that we're looking to target, we believe that ultimately an allogeneic approach will win out, certainly from a clinical and a commercial perspective. We've got two clinical stage candidates that I'll cover tonight, both addressing significant unmet medical needs, as you'll see. And over the next couple of years, we're looking to generate some very meaningful clinical trial data, and that's really where the value is in our business. So we're paid to deliver ultimately clinical data, which we hope will be positive. And if it is positive at the right stage in clinical development, those programs become very, very valuable. And we'll make our money and give our return to shareholders by licensing those programs on to the pharmaceutical companies and biotech companies that ultimately will take these through to market. It's not inconceivable that a company like ReNeuron couldn't take these therapies all the way through to market themselves, but usually for a company like us, given our limited financial resources and infrastructure, we'll look to the big boys to take those through to market and hopefully to pay a king's ransom to license the products at the right stage in their development. So just moving on to the assets themselves, we have two core assets. One is called HRPC, which stands for human retinal progenitor cell lines. Again, don't worry too much about the long kind of names here. I'll tell you what that means and how these cells work in a short while. The other one is called CTX, and again, this is a progenitor stem cell line. So ultimately, both these cell lines or cell populations have the ability to differentiate into the cell types that are lost or missing in the diseases that these patients present with. And we're targeting back of the eye retinal degeneration with our retinal cell line, specifically retinitis pigmentosa, and with our CTX neural cells, we're targeting stroke disability. And I'll come back to both of these in due course. I mentioned licensing, and we've actually already done one license deal across both of these assets. So we did this earlier this year to a company called Fosun Pharma in China. Fosun is probably in the top 2 or 3 pharma companies in China. It's a very, very substantial business. And as I'm sure some of you may be aware, it has very substantial interests beyond China and beyond healthcare. Including Wolverhampton Wanderers, Thomas Cook, of course, hit the headlines recently, and other non-healthcare assets. Importantly for us, it is very, very big in healthcare in China. So we couldn't have hoped for a better partner to help take our products forward in that territory where, of course, we don't have feet on the ground. And that deal was struck earlier this year, and we're delighted to be working with Fosun. It's one of a number of deals we'd hope to sign with pharma partners as we go forward. If there's time, I'll talk a little bit about CTX-derived exosomes, which are even more scientific and research-based, if there's time at the end. But fair to say, that's a very early stage endeavor for us, and perhaps from an investment perspective, at least for the moment, not quite as exciting as the two assets you see above. So where are we with these assets? Well, we're in phase 2 development, so mid-stage clinical development across both the HRPCs targeting retinitis pigmentosa and with the CTX cells targeting stroke disability. And as you can see, we have some interesting data readouts coming very soon, actually, in the case of RP, to build on some earlier data we announced earlier this year, from which we got quite a nice kick on the share price. Kick upwards, I should say. And the stroke study is a much larger study being run in the US, and that will read out slightly later, actually, than we'd hoped. In first half of 2021, we revised that guidance in our AGM statement a couple of weeks or so ago. Very complex study being run in a number of sites in the US, over 30 sites, actually, I think. But nonetheless, two very, very important clinical trial readouts at phase 2 within a reasonable period of time. Our exosome program is earlier stage, as I said. We're using exosomes to help other companies deliver their drugs, their advanced drugs, so it's a new delivery system we hope exosomes can help with. And we're doing that in collaboration with a number of partners, and I'll come back to that, as I said, if there's time. So just moving on to the HRPC program in a little bit more detail. So again, HRPC is an allogeneic approach to addressing retinal disease. So back of the eye retinal degeneration typically results in a loss of photoreceptors, the rods and cones in the retina that give us sight. So the aim with HRPCs is to use them as progenitor cells to implant them and to get them to differentiate into those lost photoreceptors. So thereby they're partly replacing the cells that are lost. And we also believe that they also provide trophic support to surviving photoreceptors that still remain in the retinas of patients presenting with these retinal diseases. And we do have some preclinical evidence that backs that hypothesis up. Importantly, it's all very well generating nice scientific data, either preclinically or clinically, but we've also got to be able to show that we can manufacture these cells at scale to the requisite quality standards under GMP, good manufacturing practice conditions. In other words, we can convert them to a product that can be churned out at meaningful cost of goods and therefore allows these programs to be licensed to a pharma partner that's going to want to know they can make money ultimately with these products when they're priced in the market and taken up commercially. In both cases, actually, we cryopreserve the formulation in which we deliver these cells. So once cryopreserved, these cells can be shipped and stored on a global basis with a long shelf life. So again, we're trying to get these cell-based therapies as close as we can to a standardized biopharmaceutical product, like a monoclonal or something like that, something that's readily transportable and usable at the clinical site. The first target with RHRPCs is retinitis pigmentosa. As I mentioned, this is an inherited degenerative disease of the retina. It involves a primary loss of rods and a secondary loss of cones. Typically, patients will get the disease in childhood, and it can take some years or even decades, actually, to progress. But ultimately, what you get is what you see in the image here, a gradual loss initially of peripheral vision leading ultimately to blindness. This is an orphan indication, which means it's relatively rare, although within orphan diseases, it's one of the larger ones. And essentially, it's a collection of gene deficiencies that overall go up to make the overall population of retinitis pigmentosa. As an orphan disease, we do have orphan drug designation for this in Europe and the US. And because we're taking this forward clinically in the US at the current time, we have fast-track designation from the FDA for this as well. So it helps to speed the regulatory pathway to market, all being well, for this program. Importantly, because it's a cell-based approach to a genetically mediated disease, we're not targeting just one of those gene deficiencies. We're targeting the entirety of this population. And some of you may know that there is already one approved treatment for RP for one particular gene deficiency called Luxturna, developed by Spark Therapeutic. It was approved a year or two ago. It's now been priced in the US at $850,000 per patient, but it's a very, very small patient population, no more than a few thousand patients in the US, or even worldwide, actually. So again, orphan indications are great because they're quick to market and can be priced extremely high, but that's driven by the fact that the patient population is commensurately smaller than with more conventional approaches to larger scale diseases. But nonetheless, from a commercial perspective, and of course from a patient perspective, it's a very, very worthwhile target for a cell-based approach. I'm not going to bore you with the preclinical data that we generated that got this program into the clinic. Suffice to say that at a preclinical level, using both small animal, rodent, and also large animal pig models, we could show that our HRPCs engrafted, migrated throughout the subretinal space and differentiated into those lost photoreceptors I mentioned earlier. So this gave us the evidence and the regulators the evidence when presented to enable us to go into the clinic, which is now where we now are with this program. We're currently in the phase 2A part of a phase 1/2 study. So it's a combined protocol, first of all, to show safety of the cells at ascending doses in phase 1. We've done that on the left-hand panel there. And then the right-hand panel, a further 12 subjects, or up to 12 subjects, where we are looking more for, well, obviously for both safety as well, but also to evaluate efficacy measures, looking at things like visual acuity, visual field, and so on. And this is where it got interesting for us earlier this year. By the way, the clinical trial's taking place in 2 sites in the US, at Mass Eye and Ear in Boston, and also a site in Phoenix, under Jason Commander and Praveen Dugal, who are 2 of the leading key opinion leaders in this field in the US, if not the world. So again, to get credibility for an advanced therapy program like this, you need KOL support. Okay, when investors are looking to invest in this, typically they will go to key opinion leaders and ask their opinion, right? On, what do you think of this approach? Is it viable? Does it work? What do you think? Or are they just barking? So it's very important that you get the scientific community behind what we're trying to achieve. We know that, we're on the right track, or hopefully we're on the right track, but you have to convince others. So having leading KOL support this study, for us, is incredibly important. So what have we seen so far? Just checking time, I haven't got too much time. This is very, very interesting, but early data from the first cohort of 3 patients we treated in the phase 2 part of this study. And what you're looking at here is the measure of, in blue, the treated eye, and in red, the untreated eye, patient by patient in these first 3 subjects. And what you're looking at is measuring the visual acuity over time. So obviously these data were taken at slightly different time points for the 3 patients. And you're looking at the number of letters these patients can read on an ETDRS chart on the y-axis there. So as you can see, in the case of the first patient, he got a 21-letter improvement from baseline. Patient 2 got a 25-letter improvement. Now, the obvious question you're going to ask is, well, how come the treated eye started so much lower? Well, it's actually, patients typically in this indication do present with one eye being much worse affected by the disease than the other eye. So from an ethical standpoint, of course, for a first-in-man study, you're going to target the worst affected eye in case anything goes wrong. But what you see here is some really quite dramatic improvements. The slight decline in the first few days, by the way, is purely due to the surgical procedure. These cells are injected through subretinally, which is a standard procedure if you're a retinal surgeon, and that involves a detachment of the retina. So typically in the first few days, the patient's sight will get worse. But then once that resolves and the patient recovers from the procedure itself, this is where we see these really quite dramatic improvements. And just to sort of put that in a bit more perspective, this is actually the ETDRS chart that these patients are measured on. Now, it looks somewhat prosaic, but actually it's a fairly nuanced series of tests that the patient is asked to perform when looking at this chart. It's not like you get in an optician's, a Snellen chart with the A at the top and the pyramid, if you like, that you're asked to read down. But suffice to say, any 3-line gain on this chart is indicative of meaningful clinical benefit from the patient. And that's the FDA's guidance actually for gene therapy in retinal degeneration. So what we've seen so far in these patients is on average a 4-line gain. So if we can replicate this, or at least get anywhere close to it in a sufficient number of patients, and so far in these patients it was 3 out of 3, then we're very much off to the races. This is very, very encouraging in terms of what we would want to go on to next. And the next readout on this study actually is actually next month. We have a slot at the American Academy of Ophthalmology conference in San Francisco, again, a very key conference in this area of ophthalmology, and we'll be presenting much longer-term data from the earlier patients in this study and some shorter-term data on the other patients we've treated subsequently over the course of this year. And that, I think, is going to be followed very closely for anyone interested in this particular program and in ophthalmology approaches generally to this disease. If we're successful, then obviously our next aim is to go into a phase 2B study, looking at a much larger patient population to get statistical efficacy. And we'd also like to assess RP and other indications associated with retinal degeneration as well. And there are a number of those, as you may know. I'm running out of time, so I'm just going to pit stop very briefly on CTX in stroke disability. I'm not going to get to the exosomes, but that's okay. So just a quick word on CTX. Again, these are neural cells, allogeneic, cryopreserved. We've been working with this asset for a lot longer than the retinal cells, fair to say, but we're targeting stroke, which is a much more complex and intractable disease. It's in the brain. Anything associated with the brain raises particular challenges. But again, we think we have a cell type that may work in this indication. We know what these cells do putatively. So neural cells, or certainly CTX cells specifically through our preclinical work, most if not all of which is now published. We know these cells stimulate neurogenesis, the generation of new neurons in the brain. They stimulate angiogenesis, which is increased blood flow. And we believe they also play a role in restoring synaptic connectivity in the brain, as well as modulating the inflammation or inflammatory response you get in insults like stroke. Stroke is an incredibly large unmet medical need, as I'm sure you know. Virtually everyone knows someone that suffered a stroke and maybe lives with the consequences, the disability that you get typically on one side of the body. And that's really where we're targeting CTX. Can we improve the motor deficits in these patients that survive their stroke? And the key way of measuring motor deficits, or motor deficit improvements, hopefully post-treatment, is using a measure called modified Rankin. This is where we got very good evidence in phase 2A, and it's our primary endpoint in our ongoing phase 2B study in the US that I mentioned earlier. And any one drop on this measure, and you can see in the right-hand chart here that as you go down on this measure from 5 to 0, you're less dependent, you are less affected by your disability. And you can see that pharmacoeconomically, the biggest bang for your buck is if you can reduce from MRS 4 to 3 or 3 to 2. And actually, those are the entry criteria for our phase 2B study. You have to be 4 or 3 to get into the study that we're running in the US. And these data actually were taken from a registry study done in Sweden in over 40,000 disabled stroke patients. It was published, and it's a very good illustration of the pharmacoeconomic benefit if we can get this treatment to work in these patients at a statistical level. I haven't got time to take you through the Pisces 2 study results. This was the phase 2A study that formed the basis of our ongoing phase 2B study. Suffice to say, we got a very nice response on that modified Rankin measure. It's why we chose it as the primary endpoint in the ongoing study. And in the couple of minutes I've got left before questions, this is just a quick snapshot of what we're doing now in the US in that phase 2B study. Essentially, it's 110 subjects randomized either to the treatment or to placebo sham surgery. So this is actually a placebo-controlled study, which again is incredibly challenging when you think about the fact that this treatment is actually administered by a neurosurgical intervention. But you can actually placebo control it using sham surgery, and we agreed a way to do that with the FDA, the regulator in the US, and the study's ongoing. What we're doing is treating patients between 6 and 12 months after their stroke. They have to have some residual arm movement going into the study. Again, we believe that will aid their response to the treatment. We have a number of secondary endpoints, as you can see in the panel there on the right, beyond the primary endpoint, which is at least a 1-point improvement on that modified Rankin scale you saw on the previous slide, at 6 months, measured at 6 months post-treatment. And again, we're hoping to get to a top-line readout on this study in the first half of 2021. Ignoring exosomes, it's far too scientific for the likes of me anyway. I'm just going to dip straight to the summary. So again, in summary, we are essentially a cash burn biotech business where we're developing cell-based therapeutics. Here in the UK, we have a site in Boston in the US as well. These are patentable, scalable, cost-effective therapies, we hope. And we're targeting diseases characterized by being large unmet medical needs. We have significant clinical milestones due over the next couple of years, the first one being due very, very soon. And from that, we see near to medium-term opportunities for further partnering deals beyond the one that we inked earlier this year with Fosun in China. And I think I'm out of time. I am out of time. There we go. Thank you.
Any questions, please?
Is one of your targets possibly AMG?
Might be. Yeah, so there's a number of diseases of the back of the eye associated with either the loss of photoreceptors or the loss of the support layer called the retinal pigment epithelial layer, which is I think more relevant to AMD. Again, I have to be a little bit careful because it's probably above my pay grade from a clinical perspective. But yeah, AMD is definitely a target. Dry AMD, that would be, which is where the large patient population is.
Thank you.
Do you envisage stem cell treatment being supported by the NHS in the UK?
Yes. I think pricing is going to be key. I mean, what you have to remember here is these are envisaged as one-off therapy, so a one-off administration for a lasting effect. That's the essence of cell therapy. These are supposed to be reparative treatments. So this is not a treatment you're going to get on a monthly, weekly, or even an annual basis. It's a one-shot therapy for a lasting benefit. So from a pharmacoeconomic perspective, it's reducing the reliance of those patients on health and social care systems. And therefore, when you think about pricing, it raises all kinds of issues in terms of how you price this in terms of, existing NICE models, cost per quality models that NICE typically use and other recommending bodies. And there's a lot of work going on actually among the trade bodies that we're associated with to move pricing authorities, including the NHS and NICE in particular, towards the notion of paying for these treatments, perhaps on the basis of an annuity on a pay-per-performance type basis. So you pay something up front, and if you do get the lasting effect, then you get the full price of the treatment over time. There's a number of different ways of doing that, but that's the way I think to get these treatments both adopted and paid for by national payers, including the NHS in due course. But there's still a lot of work to do to determine how these treatments are going to be priced in the future.
I've got a question for you, Michael. You're sort of concentrating on two areas with the brain disease and obviously the blind-causing diseases. Is it something that you need to concentrate on one area specifically, or are you sort of sharing your energies and your challenges across the two?
It's kind of interesting. I mean, I've been in the company long enough to sort of see both of these programs sort of develop over time. And as I mentioned, the retinal program is a lot younger actually than the neural program. I mean, the name of the company kind of gives it away, ReNeuron. I mean, we were founded on our CNS expertise.
Yeah.
I think at the moment, probably fair to say that the retinal program's sort of in the ascendancy because the early data was so intriguing earlier on this year. Mm-hmm. But of course the stroke program is now in it's in a later stage but much longer study, and there's not much we can really say about it until that study reads out. So you tend to find that programs kind of diminish in terms of regular news flow as they get into larger stage studies, and therefore other programs come to the fore because they can offer more news flow and excitement because typically they're going through an earlier stage in their development, which usually means more regular news flow. So we love all our children equally, I guess is the simple answer.
Absolutely. What is your current market cap?
Last time we looked, probably about 65 million or so. And, our cash balance, we've done quite well with cash. I mean, we have to raise money from the market. The last time we did that was 2015. So we're still living off what was a very large raise for us a few years ago, about 68 million, I think it was. But it won't be long before we'll have to consider raising more money. We'd like to try and do that, at least in part, by inking further license deals that I mentioned. But if we have to go back to the market, so be it. But we're certainly, as far as we're concerned, nearer the end of the journey in terms of value and commercialisation than we are from the start, that's for sure.
Right. And who are your main competitors in this particular sector?
Well, we're not overly encumbered by competitive threat, that's for sure. And that's partly driven by the very novel nature of what we're doing. Yeah. There are other players, and actually the more the better, frankly, because part of the challenge here is validating these treatments. And if we were the only game in town, as we were actually a good few years back, we were kind of looked upon a little bit strangely. Whereas now that the broader field has developed and you are getting cell-based treatments coming to market, that is now happening. And also incredibly valuable deals are being done with cell and gene therapy businesses through acquisition as well as partnering, then I think that's kind of raised all boats really, including ours. So I think we're now seen as a much more credible player because the field itself has moved on, and so the whole field has more credibility, certainly from a clinical and commercial perspective, than maybe it did a few years ago.
Okay, thank you.
As you have orphan drug status on the Retinitis Pigmentosa product, once you finish phase 2, does that mean you can go straight to market, or do you have to go phase 3?
No, that's to be decided with the regulatory authority in the US in particular. It's not unheard of for studies to be approved, perhaps on a limited basis, post phase 2. But it all depends on the quality of the data. But you're absolutely right. Orphan status programs typically do offer that kind of opportunity. But it's going to be driven by clinical data at the end of the day. If it's gangbusting, then we may have an opportunity. But we don't assume that.
Okay, any other more questions from the floor? No? Are you staying for a drink, Michael, to answer
Sure.
questions over drinks and canapes afterwards? Excellent.
Okay.
Thank you.
Thank you.